Hey everyone, and welcome to RUBIN'S Mind Lab, where we break down the latest research into what it really means. Ever wondered how your brain keeps track of things changing in a story, or even just in your mind's eye? Well, today we're diving into some fascinating brain science that might just give us a clue. So imagine you are reading a sentence like the chef will chop the onion and then he will smell it. Now, that onion goes from being whole to chopped, right? It is changed. But when you get to the word it at the end, your brain needs to remember which onion we're talking about and likely what state it is in. How does your brain manage to hold on to those different versions of the onion so you can follow the story? That is the big question these researchers wanted to answer. They think the answer might lie in a specific brain signal that is usually linked to visual working memory. Basically, your brain's ability to hold on to visual information for a short time. This signal is called Theta-Gamma phase-amplitude coupling, or TGPAC for short. It's a bit technical, so let's break it down. Imagine your brain activity as different waves. Theta waves are slow, and gamma waves are fast. TGPAC is when the rhythm of the slow theta waves influences the strength or amplitude of the faster gamma waves. It is like a conductor, the slow wave, telling a whole orchestra of musicians, the fast waves, when to play louder or softer. Scientists think this coordination helps different parts of your brain communicate and store information, especially in working memory. To test their idea, they did 2 experiments using EEG, which measures electrical signals in the brain from outside your head. In the first experiment, they looked at brain activity while people read sentences describing objects undergoing either minimal change, like weighing an onion, or substantial change, like chopping it. They hypothesized that if your brain is actively maintaining these different states of the object, especially when they're very different, you'd see more of this theta-gamma brain signal. In experiment 1, they reanalyzed data from previous studies. Participants read sentences and they measured TGPAC. They found the flicker of a signal, a cluster of brain activity in the left temporal region that seemed to show slightly more TGPAC for the sentences with substantial change. However, the result wasn't strong enough to be statistically significant. This meant they couldn't be sure it wasn't just a fluke. So they decided to do a second experiment to get clearer results. For experiment 2, they brought in new participants. This time, they did 2 things. First, they had people do the sentence reading task, similar to experiment 1, focusing on minimal versus substantial change. Second, and this is the key part, they also had participants perform a visual working memory task. This task was designed to reliably produce that TGPAC signal they were interested in. They used this working memory task as a localizer, a way to pinpoint the brain areas that are definitely involved in TGPAC. Then they analyzed the TGPAC specifically in those same brain areas while people were reading the sentences. And this is where it got exciting when they looked at the brain areas identified by the working memory task. They found a significant increase in TGPAC for sentences with substantial change compared to minimal change. It was like the working memory task gave them a special lens to see the signal more clearly in the language task. But they went even further. They realized that substantial change is a bit of a broad category. What if the brain signal actually changes depending on how much something changes, not just whether it changed a lot or a little? They used ratings from another study where people had scored how much an object changed in those sentences. They found that yes, the more change an object was rated as undergoing, the stronger the TGPAC signal. This is a really neat finding because it suggests the brain isn't just using a simple switch for changed or not changed, but it is sensitive to the degree of transformation. So what does all this mean? The researchers believe that this theta-gamma brain signal, TGPAC, is a crucial mechanism for how our brains maintain multiple representations of the same object, especially when those objects change states over time in a story. When you read about the chef chopping the onion, your brain, through TGPAC, is actively holding on to the image of the onion before it was chopped and its state after it was chopped. This is particularly important when the changes are significant, likely because those different states are more distinct and require more effort to keep separate. This also ties into the idea that understanding language, especially about events, might rely on the same brain processes we use for visual perception and memory. Think about it when you imagine the chopped onion. It's not that different from actually seeing it being chopped and then remembering the visual image. The researchers suggest that TGPAC might be a fundamental neural code that can represent information sequentially. Whether that information comes from your eyes or from words, it's like a common currency the brain uses for keeping track of things over time, whether they're visual objects or mental representations cued by language. They also touched on where this signal might be coming from in the brain. While it is hard to say for sure with EEG, previous studies have linked TGPAC to the hippocampus, a part of the brain important for memory and creating mental maps. This makes sense as the hippocampus is also involved in constructing scenes and remembering events. The big takeaway here is that TGPAC seems to be a common neural process that helps us maintain objects and their changing states, whether we're seeing them or reading about them. It supports the idea that our language comprehension isn't just abstract symbol manipulation, but it is deeply rooted in our sensory and perceptual experiences. When we understand a sentence about change, our brain appears to be reusing the same tools it uses to manage visual information in working memory. Now, like any study, there are limitations. The sentences they use were carefully constructed to be simple, and it's not clear if these findings would hold for more complex. Natural language also, the visual working memory task they used didn't explore how TGPAC behaves with varying similarity between object states, which is a key aspect of why some changes are harder to track than others. Future research will need to explore these nuances. So what is my take on all this? I find this research super exciting because it really bridges the gap between language and perception. The idea that the same brain activity, this theta-gamma coupling, is involved in holding onto visual information and keeping track of changing objects in sentences is incredibly powerful. It suggests a deep, underlying unity in how our brains represent and manipulate information, regardless of the source. It makes sense that if your brain is already good at remembering different visual items because of TGPAC, it leveraged that for language too. The fact that the brain signal scales with the degree of change is particularly elegant, showing a sophisticated system at play. Looking ahead, I'd love to see more research exploring how TGPAC handles more complex sentence structures or even spoken language, where the timing is even more critical, and it would be fascinating to investigate how individual differences in working memory capacity affect this TGPAC signal during sentence processing. Ultimately, this study gives us a glimpse into the sophisticated neural mechanisms that allow us to not just understand words, but to build rich, dynamic mental models of the world as described by language. If you found this dive into brain signals as cool as I did, please hit that like button, leave a comment with your thoughts on how your brain tracks changes, and subscribe to RUBIN'S Mind Lab for more explorations into the wonders of the mind. See you next time.